Welcome to Life in Biology. I'm Dr. Joel Graff, and today we're going to be working our way into Mendelian genetics. There's quite a big vocabulary to learn with Mendelian genetics, and so we'll introduce some of the terms now and then walk through an example of some crosses between different organisms with different traits. So we're going to start off with the word allele. So for each gene that's in the genome, there could be different alleles within a population. And so by that I mean that some organisms might carry the capital P version of the gene and others might carry the lowercase version of the gene, or some might carry a combination. Moving on to the next word is character. So when Gregor Mendel was working with pea plants, one of the characters was the flower color. And so the flower color would have a trait, and that trait would be either purple flowers or white flowers. So the character is the color, just whatever color the flower is. The traits are what are the possibilities for flower color. Okay? Then there's genotypes, and so then you could have two capital P's, a capital P and a lowercase p, or two lowercase p. The reason you always have two is that we are dealing with a diploid organism, and so they have two chromosomes. They have one allele of the gene on their maternal chromosome and one allele of their gene on their paternal chromosome. And so you have always pairs of genes. So a genotype almost always talks about two genes that explains which two alleles you have on your two chromosomes. Okay? So genotype is what genes are there, and phenotype is what the trait ends up being when you have a specific type of genotype. So the traits could be purple or white. If it's a capital P, so you're homozygous for capital P, that pea plant will have purple flowers. If the pea plant is heterozygous, meaning that the two are different, you've got a capital P and a lowercase p, that also leads to a purple flower. And then only when you are homozygous, both alleles are the same for lowercase p, do you end up with the white flower. And so what we're dealing with here is a trait or a character that is where a capital P is dominant to lowercase p. Really want to point out the word dominant here because we're going to come to examples of incomplete dominance, and so those will be dealt with a little bit differently. Now, one of the easiest ways to deal with Mendelian genetics is to use Punnett squares. So let's say we've got a couple parent lines of flowers. Now these are going to be pea plants. Now, these pea plants that Gregor Mendel was using were true breeding pea plants, which means that as long as you kept cross-fertilizing those plants with themselves, he had different strains where you cross-fertilize with themselves, they'd always give you the exact same traits, meaning that the only way to explain that is that you have a homozygous for that trait. And here we're going to do a parental cross where it's homozygous for the capital P allele. So that would be a purple flowering pea plant. And then here we've got lower p allele, and that's the lowercase p, and those would produce the white flowers. Because we learned about the law of segregation, we know that this first parent, let's say that's the father, can give either a capital P or a capital P. Those are the two options, so really there's only one option. So we have a capital P or a capital P here. And then the mother in this cross has two lowercase p's, and so we'll put those on this side of our Punnett square, lowercase p, and then when you have fertilization, these represent the gametes, what gene the gametes have. So this gamete fertilizes that gamete, and you end up with a diploid, because these are each haploid, and they they fertilize each other, and then you have a diploid, capital P, lowercase p. Well, we have a capital P, lowercase p is what could happen with this gamete and this gamete. This gamete and this gamete would give you a capital P, lowercase p, and this gamete and this gamete would end up giving you a capital P, lowercase p. Because that is so redundant, one thing that you can do is that you can make your Punnett square a little bit easier, because what this is saying is you've got a 50% chance of a capital P or a 50% chance of the other capital P. So really it's a 100% chance of a capital P, and then you have a 100% chance of a lowercase p, and so then you have that there. So then the new words that we're going to come to right now, the genotype, is going to be 100%, 4 for 4, or 1 out of 1, are capital P, lowercase p. And then the phenotype is 100% purple, because the capital P is dominant, the purple is dominant to the white. And so these plants, these F1 generation is what we call them, after the parental generation is the F1 generation, they're all going to be heterozygous for capital P, lowercase p, and they're all going to make purple flowers. So that generation was called the F1 generation. Well, if you take those children of that first parental cross, the F1 generation, and you cross them with each other, then we end up with the F2 generation. And so our F1 generation is a cross between a homozygous, let's say this is a male, or a heterozygous male, a heterozygous female, gametes, and then you cross those together. So there's a 50% chance it'll be a capital P and a 50% chance it'll be a lowercase p. So we put that on the axis. This line is supposed to be right down the middle, so it's a 50% chance of being on that side of the line or this side of the line. We have a capital P here and lowercase p here for the other chromosome, or for the other gamete possibilities. And so if this capital P gamete fertilizes this capital P gamete, say that's a sperm pollen and then an egg ovule, then you have capital P, capital P. If you have this lowercase p and this capital P, you would write capital P, lowercase p. It's just convention to put the capital letter first, or the capital first, if you have a heterozygous. Again, capital P, lowercase p, and then lowercase p ends up down there. So now, if we talk about the genotypes, the capital P ratio, there's 1 out of the 4 boxes is that, so I'll put a 1 there. The capital P, lowercase p, 2 boxes out of the 4 are that heterozygote, so I put a 2 there, and then lowercase p is 1. And so we would say that's a 1 to 2 to 1 ratio is how you would explain that. All right, so, or a 2 to 1 to 1, I guess would be possible too. And then a phenotype would be that there would be 3 purple and 1 white, because this capital P and capital P make a purple flower, a capital P, lowercase p makes purple, capital P, lowercase p makes purple, and then the lowercase p makes white. So that is 3 purple for every white. So out of the seeds that you would get from that cross, you'd expect 75% to be provide you with purple flowering peas, and 25% to give you white flowering peas. Okay, so this is when you first take a parental line, you're looking at 1 trait, and you have homozygous for dominant and homozygous for recessive, and you get your F1 generation, and then you go and you take the F1 generation and cross it with itself. There is a term for that that's called a monohybrid cross. And so I'll maybe make a video about a dihybrid cross at some point, but this is a monohybrid cross because we're mono for just 1 trait that you're dealing with. All right, well, there's your introduction to Mendelian genetics. I'll talk to you later. Like and subscribe or not.